Hello and welcome to ResearchPod. Thank you for listening and joining us today. Understanding how living cells work is difficult due to the number of varied and complex processes occurring within them. This complexity can be clarified by breaking these processes down into simpler components and focusing on a particular mechanism. One approach is to use mathematical equations, the basis of computational modeling. Dr. Susan Mertins, the founder and CEO of Biosystem strategies in the USA, explores how ordinary differential equations and machine learning can be applied to cancer data for biomarker discovery and drug development, leading to improvements in personalised medicine. Biology is the study of living organisms, which can be described as complex systems. Cells can have many different and sophisticated functions, for example, protein production and photosynthesis. But because biological systems are dynamic and convoluted, this makes them challenging to analyse. However, specific mechanisms can be interpreted more easily using quantitative metrics. In other words, cell behaviours can be simplified by focusing on one specific aspect and only considering cell elements relevant to the question being asked. The biological phenomenon can then be approximated using mathematical equations. This is the principle underlying computational modelling, where biological mechanisms are represented using laws from physics and chemistry. Computational modelling enables us to simulate reality and make useful predictions in medicine, from drug development to biomarker discovery. Dr. Susan Mertins, founder and CEO of Biosystem strategies, uses both computational modelling and machine learning to detect drug targets and biomarkers that will help develop personalised approaches to cancer treatment. The first step in computational modelling is knowing what you want to model. This may seem obvious, but being clear about this determines the choice of model and analysis. Following this conceptual phase, the actual model is constructed, and the code for the simulation is written. While doing so, it's important to consider how the model can be verified by estimating the parameters and calibrating the model. As Musuamba and colleagues describe in a 2021 paper, verification can be seen as solving the equation in the right way, while the following step, validation, consists of solving the right equation. Model validation is the ability to simulate the conditions of interest with certain sensitivities and uncertainties, which reflect the quality of the model. Finally, the simulation is compared with comparator data to assess the credibility of the model, for example, using the results of a lab experiment. The type of model needed depends on the spatiotemporal scale of the biological process. For example, a reaction between enzymes will be faster than the development of a whole organ. The intracellular scale can be described with ordinary differential equations, or ODEs, and involve a single variable that can describe a particle moving through time. Mechanisms, such as protein gradients, however, are described by partial differential equations, or PDE, which involve multiple variables and can therefore describe a surface changing over time. At the cellular scale, there are two main options of models, depending on whether space and time are considered discrete, known as on-lattice, or continuous, known as off-lattice. Agent-based models are a typical off-lattice model, used to describe the interactions between individual elements of the system. On-lattice models can be separated into two main categories: cellular automata, whereby agents are represented by a single pixel, and cellular POTS models where agents are a collection of pixels. In other words, collective as well as individual cell behaviour can be simulated. Cellular POTS models can therefore provide a greater resolution, but are more computationally expensive. Whole cell modelling can be used to incorporate the different scales of a biological system, and consequently predict how observable traits will arise from genetic mechanisms. Cancer is a complex disease, which is hard to treat because of its heterogeneity. Different patients have tumours with different mutations, and even cells from the same tumour can have different mutations. Treating a tumour is therefore challenging because if some cells are resistant to a treatment, the tumour can recur. Patients also have different metabolisms, so react to each drug differently. Understanding the actions of the disease at a cellular level is critical for effective treatment. Cellular responses are the result of a series of molecular events, and together are referred to as signal transduction pathways or reaction networks. The shape of proteins determines the outcome of the pathway, as shape dictates which proteins are attracted or repelled and how proteins interact with each other. These transduction pathways depend on the concentrations of proteins and the speed of their reaction. They control various mechanisms such as gene expression and metabolism, which cancer interferes with. Output from the reactions also depend on feedback loops and are sensitive to input as well. Specifically, cellular responses can be inhibited through negative feedback loops and promoted via positive feedback loops. Under normal conditions, doubling the amount of proteins doubles the reaction rate, but ODEs, representing protein concentrations over time, have revealed that chemical responses are not always linear. For example, in a saturated solution, where the concentration of a protein has reached a certain value, the reaction rate in the equation changes, and the initial protein concentration is not proportional to the output cellular response. Think of motility or energy production, for example. Instead, the protein becomes ultrasensitive, meaning that the pathway output is more affected by small changes in the initial protein concentration. Modeling signal transduction with computers, therefore, can be described as dynamic or kinetic, and using these models allows the study of drug mechanism of action or pharmacodynamics. The mitogen-activated protein kinase or MAPK pathway involves proteins called kinases, which add phosphate groups to other proteins to modify their function. This phosphorylation mechanism is possible only when the protein is activated, meaning that it is in the right shape. This cascade includes proteins which are mutated in cancer, as they control cell proliferation, cell death, cell motility, and differentiation. In a 2016 paper, Rauch et al. Explained how computational modeling helps make sense of the MAPK network. For example, when two proteins react and form a dimer, two identical molecules link together, a process known as dimerization, this can lead to drug resistance. Overall, the efficacy of the drug depends on how the binding of the drug and the protein impacts the shape of the protein. Dimerization tends to be favored between a drug-bound protein and a drug-free protein, but this conformation activates the drug-free protein instead of inhibiting it. You can compare this dimerization process to a puzzle. If the two pieces have a complementary fit, they have more chances of being stable together. In this case, the drug-bound protein and the drug-free protein bind easily together, leading to the accumulation of such dimers and consequently to drug resistance. Nonetheless, two drugs ineffective on their own can overcome drug resistance if they are combined. In her review, Mertins illustrates how computational modeling is used to represent protein concentrations of the MAPK kinase pathway. She explains that an ODE can be used to describe the amount of protein modification, also known as the amount of phosphorylation. This simulation requires an input from protein databases, known as proteomic databases, to set the initial quantitative parameters. From the ODE output, machine learning can be applied to discover novel biomarkers and targets for new drugs. Indeed, machine learning helps us make connections that are unlikely to be made by humans, such as predicting cancer survival or being able to detect cancer early. For instance, the extent of protein modification resulting from the ODE can be investigated to find a correlation with cancer prognosis. Besides, novel drug targets can be found by mimicking mutations which prevent protein production or function. Parameters representing proteins can be removed to simulate the effect of such mutations, or parameters can be adjusted to slow down function attributed to its activity. By doing so in a systematic way, the importance of proteins can be assessed by analyzing the outcome of the simulation. If the absence of a protein results in cancer cell death in the model, it suggests that this protein is a promising drug target. There are many advantages to using computational simulations. They are usually faster and cheaper than lab experiments, enabling more candidate drug targets to be revealed, significantly helping to tailor medicines to individual patients. Computational modelling also decreases treatment-related risks. Digital twins can be made to understand how the cancer would react to a drug depending on the molecular profile of a patient in a simulation. Moreover, it can help predict drug resistance and the evolution of a tumour. Conditions and parameters can be set to answer a specific question, and the time steps can be controlled to focus on a specific aspect of interest. Of course, simulations are often constrained by assumptions, which means they are a simplification of reality, and the results do need to be validated in the real world. Despite these limitations, however, computational modelling enables us to move much more quickly and efficiently towards drug discovery and personalised treatments for complex diseases. That's all for this episode. Thanks for listening. And stay subscribed to ResearchPod for more of the latest science. See you again soon.